Welcome to Lab Notes. I'm Joe Fetterman with Colliers Life Science Advisors, and we're here with Chris Hogan, the Senior District Sales Manager for Pennsylvania for Waters. And Chris, we're delighted to have you. Chris, you're a hometown native of the Philadelphia region and a very appropriate guy to be leading the charge for Waters in the region. Welcome to Lab Notes.
Thank you, Joe. I appreciate the time today to spend with you to discuss Waters Technologies.
So, Chris, how did you get into doing technical sales in the life sciences for equipment and instrumentation?
It started off, I graduated with a degree in chemistry from Bloomsburg University in 1996, and I migrated to a bench chemist position at a company called Johnson Matthey in King of Prussia. From there, I took another role with the Dionix Corporation as an applications chemist supporting field. From there, I then migrated into a sales position and have been with Waters for the past 14 years doing just that. I recently took a role as a district sales manager for the Pennsylvania district with Waters.
So, Chris, one of the interesting things to me about Waters is that you guys are really focused on instrumentation for cell and gene therapy and very much on the leading edge of innovation in that area. Let's talk a little bit about some of the initiatives that you have going on, particularly that with Megadalton.
We are developing a various array of applications and products aimed at helping this market, especially with those requiring mass measurement.
And let's talk a little bit about that in terms of what that means. So, we've had a previous conversation. We talked a little bit about the investigation of large molecule and both the state of the world previously and the innovation that you all are bringing to how bench scientists can look at large molecules in a different way.
Okay, so Waters has an array of products that are aimed at doing qualitative and quantitative analysis. HPLC UV, or HPLC optical detection, I should say, along the lines of also having conventional mass spectrometry. And then even we have a more collaborative effort these days with Megadalton Solutions using charge detection mass spectrometry to see very large molecules that conventional mass spectrometry can't see. Conventional mass spectrometry can measure the constituents of some of these molecules, but we can now, with this collaborative effort, we can measure the molecules in their intact state.
And Chris, this is no small matter. This is a meaningful breakthrough. What does that mean to an individual scientist in terms of their process and the time in really studying the molecule as developed?
A lot of the tools that exist these days cannot measure the mass of an intact molecule that is quite this large. This essentially means to some of these organizations that they really get a good snapshot, a good picture of the molecule that they see, or that the molecule that they are producing. They are able to characterize it much faster, and they're able to see it and be able to reproduce it and tweak the molecule much more quickly than some of the analytical tests that exist today.
Good. And one of the other innovations that you folks have developed is something that you call Immerse Cambridge.
Correct.
And here in Philadelphia, we're sad to know that it's Immerse, that Cambridge is part of that name. But tell us about Immerse. Why have you done it? What's the purpose, and what happens at Immerse Cambridge?
Okay, that's a very good question. So, Immerse Cambridge is a collaborative platform for Waters to try and acquire maybe difficult assays, molecules that Some of these organizations are trying to analyze. Maybe they don't have a means to do so. So we offer up a collaborative environment for some of our, cutting edge technologies, BioAccord mass spectrometry, the CDMS, some of our UPLC technology, and some of our latest and greatest mass specs to analyze molecules in this what I call a collaborative environment, so that we can showcase our products, but also help them understand what their molecules look like.
So that facility's been open for about a year now, Chris?
Slightly over a year, that is correct. There had been some delays, of course, due to the pandemic, but we were off to a very strong start in 2021 in that environment. Not to be confused, we do have field offices, that exist here in Pennsylvania. We do have a field office in Plymouth Meeting with some of our conventional mass spectrometry and our HPLC and UPLC technology as well. But for some of more of our cutting edge tools, we do promote Immerse as the platform to assist. Because some of it can be very specialized in the needs of our customers, but also in the requirements needed for scientific interpretation.
Chris, I appreciate that you mentioned the Plymouth Meeting facility, and because it really underscores the client base that you have in Philadelphia is pretty vast. Can we take a quick minute just to review that a little?
Sure. Waters has clientele from academia through government and within industry as well. We do service all of the schools within the area: Penn, Drexel, Thomas Jefferson, Temple. Equipment exists in all of those academic centers, but we also are very much embedded with startup cell and gene therapy companies, established pharmaceutical companies. And even beyond that, we actually have a very notable, outside of Philadelphia, collaboration with BioNTech for the COVID vaccine.
So I think one of the things that is most interesting in our conversation, Chris, has been, with your history of working on the technical side, and then making the decision that to expand and get out and really have a more interactive role. I'm very much interested in how you begin the process with a new client, and how you both get an understanding of where they're trying to get and how that fits your platform and how you deliver solutions for clients.
It's an excellent question and a very important one, because we just don't sell equipment to somebody and hope that they will use it. Really, it is an iterative process where we must have a conversation to understand their needs, their molecules, what they're trying to accomplish. Each lab can be very unique within the needs of the molecules that they're trying to produce, characterize, or even release to the general public. So when we visit and have conversations with scientists, whether virtually or live, we first have a meet and greet, establish rapport, but then also establish exactly what it is that they want to do and determine whether or not any of our products will suit their needs. So after we have an initial discussion, we typically will demonstrate our capabilities. And that could be from reference material that they can provide us, if they need to stay away from any proprietary material that they have. But if they can offer us up sample sets, we are more than happy to do trial runs within our respective laboratories, whether it be Plymouth Meeting or Immerse Cambridge, to then show our clientele what is able to be accomplished on our equipment. From there, that can help them justify whether or not our equipment is appropriate for their needs, or whether or not we need to do a second set of tests because it needs to be refined.
Well, that's a great story. So, Chris, as we wrap, I think I can't help but ask you the question. From the Plymouth Meeting sales office and the capability that you have there, when's Immerse Philly coming?
That is a question that will need to be asked to our senior management. I think in understanding the level of science that goes on within the Philadelphia area and the cell and gene therapy companies that are emerging within the area, that is something that we need to work on as an organization.
All right. Well, we'll help you cultivate the case for Philly. I'm looking forward to Immerse Philly, and can't wait to see you down in University City with presence there. Well, Chris, it's great to be with you. We really appreciate a look at some of the innovation that's happening at Waters, and really a dive into the technical side of instrumentation and the role that you folks at Waters are playing in advancing gene and cell therapy. Thank you very much.
Joe, thank you very much for your time.
You bet. Bye-bye.
Bye-bye.